Can you guys hear me okay? Fabulous. Thanks everyone for being here for my talk. I know it's the last day of the conference and everybody's kind of tired of it. Luckily, I'm not giving a talk on evolutionary biology. I barely believe in evolution itself, so you know. I'm actually a cell biologist. So let's get started. So, two of the most known treatments for cancer are chemotherapy, radiotherapy, but a lesser known treatment is hyperthermia. Basically, hyperthermia is a selected targeted heat towards a tumor of 42 degrees and above, and it's usually given in order to induce necrosis, kill the tumor. It's also given as an adjuvant to chemotherapy and radiotherapy because it sensitizes the tumor towards those treatments. However, it has to be spaced out in time in order to avoid the acquisition of transient thermotolerance, which is basically fancy words to say we space it out so the tumors don't start resisting. There's a lot of research going on in hyperthermia, and this is basically just to point out that the mechanistic explanations towards this phenomenon are still quite unclear because it's very broad and can involve a lot of pathways. In our lab, we believe that thermotolerance is thought to be mediated by what we call the cellular adaptive response. That's just a fancy word to say that the cell sees some mild stress, the cell upregulates defenses, and so when a severe stress comes afterwards, the cell is ready to kick its ass. And so we know that sublethal environmental stress can activate the cellular adaptive response, and it can pass by multiple pathways such as upregulation of antioxidant defenses, autophagy, accumulation of heat shock proteins, etc. As I said, it improves survival towards lethal stress, but the mechanistic explanation of the broad phenomenon still have much to be desired. In our lab, we work on ROS and NRF2, and so when we talk about stress, here we can't really bypass the big boogeyman of ROS. Reactive oxygen species, or ROS, its effect is concentration dependent. At low or moderate concentration, it affects multiple things such as cell signaling, proliferation, immune response, and I could stay here all day naming other things. Some people might even say that its effect is a bit pervasive. But at high concentrations, it can induce oxidative stress, damaging lipid membranes, DNA, proteins, and then accumulating insults and then eventually causing death in its host or in the cell. One of the proteins that are of key interest to us is NRF2. This is the master regulator of the antioxidant response and is directly impacted by ROS signaling. Basically, if you look, ROS will basically start oxidizing the cell, and when the environment is prolific enough, it will allow the phosphorylation of NRF2, which dissociates from its host keep1 and then translocate to the nucleus, allowing it to bind to antioxidant response element, and the transcription of cytoprotective gene goes on and on and on. The interplay between ROS and NRF2, we know that it's there, but it's such a broad phenomenon, and ROS can occur from over 40 different sources in the cell. So basically pinning it down and understanding the intricacies of this is still an objective we have in the laboratory in regards to cell stress and acquisition of thermotolerance. So basically, the objective of my study here is three-part. The first part was to evaluate the impact of the cell adaptive response on cell survival when faced with lethal stress. Secondly, we wanted to look at the impact of NRF2 and its role in cell defenses during this lethal stress. And finally, we wanted to propose at least the blueprint for a mechanistic explanation to the acquisition of transient thermotolerance in regards to hyperthermia and cancer treatments. So our hypothesis is pretty simple here. We believe that the transcription factor NRF2 is critical to the acquisition and maintenance of thermotolerance, and we believe that actually ROS from several different sources, such as mitochondria and ADPH oxidase, are essential as precursor signaling molecules in order to activate this thermotolerance. However, when we tested this in the laboratory, only one of these sources turned out essential. Considering the session you guys are in and the title of my slide, I'm gonna let you guess which one it is. It was mitochondria. So for our model of study, we used HeLa cells, which are basically transformed epithelial cells and are the most well-studied cell line in humans. I say humans because they're transformed now, but yeah, you guys get it. So we took these cells and we shocked them at 42 degrees for 3 hours. This is basically a mirror of the hyperthermia effect where we're going to shock and try to kill these cells with a lethal heat of 42 and above. And for other cells, we took the same ones and we pre-treated them beforehand at 40 degrees for 3 hours. Why 40 degrees? Well, it's been shown by our laboratory that a pre-treatment of mild heat of 40 can induce the acquisition of thermotolerance and can protect against subsequent lethal heat stress. So we're basically using this as a model for the acquisition of thermotolerance in cancer cells. We then evaluated those cells via confocal microscopy for ROS analysis, flow cytometry for ROS and apoptosis, spectrophlorimetry for enzymatic assays of caspases and whatnot, and then Western blotting for levels of key protein. So what did we find? Well, firstly, if you look here in A, we have levels of DHE, dihydroethidium, which is a probe that tags cellular superoxide. So when you're going to be seeing DHE for the rest of the presentation, just take it as cell superoxide. And over 3 hours, there's significant increase of ROS during the stress. It's kind of be expected. You're stressing the cell, you're killing it over it, and there's a lot of ROS. We also looked at levels of hydrogen peroxide, which have a more acute increase here in B, but maintain levels that are pretty normal, or not normal, but pretty stable over the next 2 hours. So when there was such a big increase in ROS, we decided to look at activity of caspases. So we looked at activity of caspase 9 and 3, which are respectively an initiator and executor caspase. And we noticed that there was a significant increase of both of those enzymes, leading us to surmise that, at 42 degrees, cells are dying and ROS are killing them. And then I mentioned NRF2, which is the protein of key interest in our lab. We evaluated its levels over those 3 hours and noticed that there was a significant increase of approximately 45 to 50%. So whatever was happening, we knew that NRF2 was implicated. So what we decided to do after is we created knockdown and overexpressor cell lines for NRF2, knockdown because the knockouts were not viable in our laboratory, and then we evaluated cell death during the same heat shock of 42 degrees. And you'll notice that we have our wild types here in black, knockdowns in red, and then overexpressors in green. And when we knock down NRF2, we're basically increasing the amount of caspase activity that's happening, and so decreasing cell survival. Vice versa, when we overexpress NRF2, we have a decrease of caspase activity, which is almost flatlining, and thus an increase of cell survival. So it seemed that NRF2 here had a critical role in determining the fate of the cell during the heat stress. We looked at ROS signaling afterwards to see whether or not there could be a pattern that was involved in this phenomenon. And quite similarly, wild-type cells had less ROS than the knockdowns, and overexpressor cells had less ROS than the wild-type. So there seems to be a pattern where there is an intimate link between the ROS levels, NRF2, and cell survival during the heat shock at 42 degrees. Then what we did is we took these cells and we did the pretreatment of 40 degrees for 3 hours, and we evaluated ROS levels and caspase levels. So in A, we have ROS levels by confocal microscopy, and then in B, we have ROS levels by flow cytometry, just to really eliminate any potential bias of the methodology. And you'll notice that although in the beginning there's higher levels of ROS in the thermotolerant cells in blue, which is normal because they've seen 3 hours of heat, at the end, there's almost no increase in these cells, while control cells have significant increases of ROS that are almost 100% higher. We looked at caspase activity as well in C, and you'll notice that there's almost no increase of caspase activity relative to its original level after the thermotolerant treatment of 40 degrees, while again, wild-type cells have significant increase of caspase activity. So not only was thermotolerance here reducing levels of ROS at the end, it was also inhibiting the activation of high levels of caspase and apoptosis. So I extracted the data here from this other model here in D, where the wild type is just a referential frame over here. We have our thermotolerant cells here that were in blue. And then what we did is we took our knockdown and overexpressor lines, and we also did the pretreatment to see whether or not the presence or absence of this protein could inhibit or otherwise elicit a better response in terms of cell survival. And what we see here is, again, the knockdown cells have higher levels of caspase activity, while overexpressor cells have lower levels of caspase activity. So the pattern is consistent with the effect that NRF2 is critical in mediating here the acquisition of thermotolerance. But the question remains, well, here are the ROS levels that we also evaluated, and it's the same pattern that we see as shown before, where knockdown cells higher, overexpressor cells lower. But now the question remains, what is the signal coming from? What is activating NRF2? What is giving it the go to say, all right, let's upregulate these defenses, let's make sure the cell is survival. So In a lot of literature, we've seen that ROS can have pervasive effects, but we believe here that they were acting as potential signaling molecules when pretreated at 40 degrees, and we're basically telling NRF2 to go wild, upregulate, and then upregulate a host of cellular antioxidant defenses. So we evaluated the contribution of ROS sources from multiple different sources: NADPH oxidase, xanthine oxidase, mitochondria, and I'm not going to stay here and name all the sources, but what we found was the most interesting source of ROS that could affect the cell in terms of survival was from mitochondria. So using a selective mitochondrial antioxidant, MitoQ, which is basically translocates to the mitochondria, enhances the activity of coenzyme Q10, and also acts as a selective quencher of mitochondrial ROS. We treated these cells beforehand with MitoQ, and then did the 40-degree treatment for 3 hours, and then exposed them to 42 degrees. So we have our wild-type cells here in black, our thermotolerant cells here in blue, and the thermotolerant cells treated with mitochondria in red. And although they had less levels of ROS at 40 degrees, which is not shown here, during the severe heat stress, they had no significant difference of loss levels relative to the wild type. So we had here an idea where mitochondrial ROS could have an important effect in terms of cell survival, and not just ROS in general. So we evaluated caspase activity again to see if that translated to cell survival, not just ROS levels. And lo and behold, control cells and thermotolerant cells treated with MitoQ had no significant difference in caspase activity, while thermotolerant cells were significantly lower. So what we're seeing here is an inhibition of a certain signal that could give the go-ahead for NRF2 to activate cell defenses and mediate cell survival. I want to leave you here with a final piece of candy, which is a bit independent from what I've done here, but still quite important. In cell, NRF2 and KEAP1 are usually in a heterodimer, but there is a third protein, PGAM5, which can tether it into a ternary complex, bind it to the mitochondria, and act as a mitochondrial redox sensor. You can see here, neatly tucked between the mitochondria. Moreover, PGAM5 is also the convergent pathway of multiple necrotic and caspase-independent apoptosis pathways. So the presence of this protein during a severe heat stress can be indicative of cell death that is independent from caspase activation. We evaluated levels of PGAM5 in control cells, thermotolerant cells, and thermotolerant cells with MitoQ, and noticed that thermotolerant cells had 50% less levels of PGAM5 already at the basal level. So it was reducing this pro-apoptotic protein at significant levels. And while there's a trend that seems to be in the reversal for MitoQ treatments, it's not significantly different from the thermotolerant cells, so I'm not going to comment on that. However, what I will say is that after 3 hours of severe heat stress, the only significant difference in PGAM5 levels was between control cells and thermotolerant cells. So, summarily, what we've seen here is that there was an oxidative stress at 42 degrees that increased NRF2 levels in apoptosis in a dependent way. When we knocked down NRF2, we had increased ROS and decreased cell survival, and vice versa. The thermotolerance effect that has been shown here seems to be NRF2 dependent, where its presence can amplify the effect of thermotolerance. Fourthly, pretreating with a selective mitochondrial antioxidant seems to reverse the acquisition of thermotolerance, indicating that it is a crucial signal in the activation cascade here. And finally, cell survival may be affected by levels of PGAM5 independently of caspase activity in the cell. So in conclusion, thermotolerance seems to require a two-key activation system that is mediated not only by NRF2 and general ROS, but specific ROS that are come from the mitochondria. And our work sheds light on the phenomenon of thermotolerance and provides a blueprint for potential therapeutic targets to bypass the acquisition of thermotolerance in the context of hyperthermia treatment and anti-cancer treatments. Thank you very much. Pierre? Yes. We didn't evaluate complex activity. We had plans, but unfortunately 5 years wasn't enough, believe it or not. But if I were to do it all over again, I would love to evaluate mitochondrial complex activity and give you an answer. I'm sorry, I can't speculate on that. Dan? Thank you.
Okay, today we're going to talk very quickly about intergenomic conflicts in a context of a never-changing climate. So, in Flo's lab and Nick Lane labs, we are really interested in mitochondria and bioenergetics in general, and mostly in the evolutionary dynamics of the mitochondrial DNA and in the phenotypic effect of non-neutral mitochondrial DNA variation. So mitochondria, these tiny bean-shaped organelles producing most of the energy we need for living, they do so using several proteic complexes forming the electron transport chain plus ATP synthase, and they have their own in-house genome, the mitochondrial DNA, which codes for code subunits of some of these complexes. However, the tricky part is that most of the genes needed for mitochondria to function comes from the nuclear genome, and the products are important to mitochondria. So the proteic complexes forming the electron transport chain are chimeric units formed by subunits coming from two different genomes. So there is a need of coevolution between the two genomes, selection for genomic match. Fail to do so, and we could have some problems, as exemplified in many organisms, from yeast to multicellular eukaryotes. Mitonuclear mismatches might result in hybrid breakdown. The best example that we have is Ron's copepods, in which mitonuclear mismatch produces a strong hybrid breakdown at the level of F2, mitonuclear dysregulation, an impact on fitness, and so on and so forth. Okay, so our climate is changing, so prediction for the coming decades includes an increase in temperature, in its variability, change in dietary regime, and so on, change in the distribution of populations, and all these factors are predicted to place a large burden on mitochondrial function. Well, temperature and diet, we know, but also the distribution of populations. If we think about allopatric populations, for example, locally adapted to different environment, admixture, so secondary contact, overlap, admixture might result in mitonuclear mismatch if the mitonuclear set of genes were incompatible from the beginning. So we focus on insects. Why? Because they are the foundation of trophic networks, they are important pollinators, and we know that hybrid breakdown might be widespread in insect populations, so we want to check whether mitonuclear interactions how far could they contribute to the current crisis, because we have a drastic decline in insect populations. We have seen that in the last 15 years at least. So here, we use Drosophila as a model. We go to Australia for that. We use the Australian cline, which is defined by a predicted variation in genotype and phenotype. So we have different populations. We have two main mitochondrial haplotypes, the T, which is the red one in this case, and the M, which is the blue one, whose frequency changes across, depending on the latitude. So in northern population, tropical population, we have a prevalence of the T haplotype, whereas in southern population, the M1. The two haplotypes are defined by 15 SNPs, widespread along all the mitochondrial genes, and all synonymous mutations. And there are some evidence that they might contribute, these two haplotypes, to local adaptation in these flies. Phlo found out that the two haplotypes confer either heat or cold tolerance in these species. So northern population, tropical, so the T haplotypes confer heat tolerance for them, whereas in the south, M haplotype confers more cold tolerance. So here is our mitonuclear panel. So using balancer chromosome and a crossing scheme, we produce 4 different lines, reproducing what we see in the wild. So we have on the left 2 putatively matched lines, TT and MM. So the lowercase letter is the mitochondrial haplotypes, the uppercase letter refers to the genome, so the nuclear genome. And so we have two matched populations, and on the right we have two putatively mismatched hybrid lines where we mingle with the genomes. And yes, and they represent the predominant and the rarer mitonuclear combinations that we found either in the northern or southern population. Thanks to Damien for producing the lines, our collaborator in Australia. So our predictions are pretty straightforward. We expect to see signatures of coevolutions between mitonuclear genes, whereby the predominant lines of each northern or southern population will perform better than the rarer combinations. And we test a bunch of phenotypic traits from mitochondrial physiology to life history traits to we track also the frequency of haplotypes with experimental evolution. Finlay is doing so. And yeah, and we test a bunch of different, we test like genetic and ecological stressor, like mitochondrial combination, sex, diet, temperature change, and so on and so forth. So, do mitochondrial interactions impact the phenotype? Yes, we have previous evidence on these flies, recently published, at standard conditions, so no variation in temperature, nor in diet. That's it, so we just tested mitochondrial combination and sex, and these are just two highlights, but long story short, we see that both hybrids, which are the yellow and the green ones, if you see in the graphs, have higher levels of mitochondrial respirations, which is widespread across all the parameters, most of the parameters that we see, but they also suffer a decrease in fitness, decrease in reproductive success, slower development, and lower survival, and so on. So yes, there is an impact, a sort of trade-off between mitochondrial bioenergetics and fitness. So here I will present some preliminary data that we have with, we added temperature to the equation. And so we start with mitochondrial physiology and ROS. I selected a few graphs here. So on the top left, we have the mitochondrial respiration sustained by complex 1. On the right, we have principal component. So basically, it's a summary of all the mitochondrial physiology parameters all together. So it's kind of a summary of all mitochondrial functioning. And on the bottom, we have ROS production. Long story short, what we can see, the plots with the solid color refers to standard temperature, whereas the dash ones refers to high temperature ones. And we have females and males, and the colors refers to the lines. So we have the red and the blue one are the match ones, and in the mid we have the mismatch lines, okay, with the temperature treatment. So as we can see, as we saw before, standard condition mismatch lines perform better, or they have higher respiration rates. Whatever is better is not really clear. But when we treat them with high temperature, they suffer a steep decrease in mitochondrial respiration, mostly localized at the level of complex 1. In both females and males. But ROS is not really impacted. So a working hypothesis is that they maintain kind of ROS homeostasis by decreasing their mitochondrial respiration, might be. Then we test metabolic rates, so whole organism CO2 release, basically in this case. On the top we have females, on the bottom we have males. On the left, the solid one are 25, and the dash bar and dash plots refers to high temperature. So as we saw for mitochondrial physiology, there is an increase in metabolic rate as standard condition, but this flats down when we increase the temperature, the same as we have more or less for mitochondrial physiology. Development, nothing is really happening. In general, mismatch lines develop slower than the other ones. And when we treat them with high temperature, the only effect is on the cold-adapted match line. I go fast because I don't have much time. Then the last three parameters. So here we have reproductive success. On the left we have female reproductive success, fecundity. On the right, we have the male one. Okay, as we can see, according to previous data, we have a decrease in fitness in hybrids, so in the yellow and in the green one, at standard temperature, as we saw before. But when we stress them with high temperature, they increase their reproductive success, both females and males. Also, the standard lines are doing so, but more or less, there is no decrease in fitness. Last but not least, we go with longevity, survival. And on the top we have the females, on the bottom we have the males. The dots refer to standard conditions, standard temperature, whereas the other one will be the 29, the one with the shorter lifespan. So as we can see here, hybrids really die faster than the other ones. So this is a clear, pretty clear result in terms of life history trade-off. Okay, so increase reproductive success at the expenses of longevity. So resource allocation towards reproduction instead of somatic maintenance, I will say. So these are the preliminary results. Happy to discuss about them with you guys. Okay, so take-home message is that yes, mitochondrial interaction have a huge impact on phenotype, and this is pretty consistent in our mismatch and match lines. And so it is important to include these factors, so mitochondrial interactions, into future studies where we want to predict the capacity of natural population to adapt to an ever-changing climate. Thank you very much to Flo and Nick Lab for the opportunity for this beautiful research. Thank you very much. Please.
Yeah, really awesome study, and I love how you looked at all these outputs across organizational levels.
Yeah.
Any thoughts on sort of how these mitochondrial bioenergetic things that you measure, relate to phenotypes at higher levels like metabolic rate and ultimately fitness? I feel like that's something that's a bit muddy in the literature, and you kind of alluded to that. And it strikes me
Yeah.
as a really awesome way to look at that.
Yeah, so we would have expected like decrease in fitness to be linked with a drop in mitochondrial respiration, for example, and metabolic rate. We find the opposite. However, what is a good phenotype, mitochondrial phenotype, and what is bad? We still don't know. So the higher respiration rates that we have might be linked with dysregulation or overcompensation for mitochondria that do not work well. And also, I didn't mention that, but mitochondrial physiology has been tested in thorax, so flight, muscle, and so on. And we can have different phenotypes in abdomen or so. We should test also mitochondrial physiology in gonads and so on to really see if they match, let's say, the phenotype. Yeah. Thank you.
Cool. Hi, everyone. Thanks for coming on the last morning of the conference. My name is Phred Benham. I'm currently a postdoc at the University of Massachusetts. I'm excited to talk about some work I started as a postdoc at the Museum of Vertebrate Zoology with my co-authors Jennifer Walsh, Kevin Winker, and Rory Bowie. So if you saw the previous talk, you're well primed for this one. So we know that most eukaryotes have two genomes, one, the small little mitochondrial genome, which was captured from a bacteria, and the larger nuclear genome. And these two genomes interact in many key organismal functions, especially oxidative phosphorylation, where you have both mitochondrial encoded proteins and nuclear encoded proteins that interact very closely in things like the cytochrome oxidase complex you see here. And this really sets the stage for coevolution and conflict between these two genomes, which can play out in different ways among different species. So in species that have been diverging in isolation for a long time, if you bring them into secondary contact, you have a number of examples like Antigriopus copepods and Giphophorus swordtail fish, where there are actually mitonuclear incompatibilities that can severely impact fitness of the hybrids. However, this is by no means the only outcome of secondary contact between divergent mitonuclear co-evolving lineages. And we often see mitochondrial genomes jumping across species boundaries in cases like the golden crowned sparrow or I think that's a rufous-tailed chipmunk. And so understanding the evolutionary processes that lead to either introgression or incompatibility can be really important for understanding how these mitonuclear interactions can contribute to speciation and adaptation. So in the case of mitochondrial introgression, there's a number of hypotheses put forth about how this can happen. So you can have adaptive introgression, like in the instance where a species colonizes a new habitat and receives the mitochondrial genome from a species already adapted there, like high elevation siskins. Alternatively, in small populations, you could have the accumulation of deleterious mutations, and new mitogenome may actually improve fitness if you swap it out with the junk mitogenome. And finally, you can actually have strong purifying selection on the mitogenome, which would preserve its ability to interact with divergent nuclear genomes. So in this Drosophila paper, they're actually able to introduce a mitogenome from one species onto the nuclear background of another without any fitness consequences or observable fitness consequences. So I think there's a lot of work to do to understand how these different possibilities play out in nature, and I think we need more empirical work. And I think a real golden opportunity here is these cases where we've found divergent and sympatric mitochondrial lineages in the absence of any sort of nuclear structure. So a great example is this Malagasy songbird, where you have 4 mitochondrial lineages up to about 7.5% divergent from one another, but no nuclear structure. And so while this pattern's fairly rare, it pops up all over the place on the tree of life. So everything from tuataras to earthworms and fig wasps and honey guides. And the reason I'm interested in mitonuclear adaptation is because it shows up in the coolest North American bird, the savanna sparrow. So this is one of the most widespread North American songbirds. It's breeding in open grasslands, basically from extreme northern Alaska all the way down into the Guatemalan highlands, found in a wide range of different habitats. And importantly for this talk, early phylogeography work using the ND2 gene of mitochondrial gene showed that there were 3 main mitochondrial lineages. You have this purple lineage that is primarily found in northwestern Mexico and largely isolated from the other 2. But then you have this A and B clades shown in red and blue, which, as you can see in these pie charts, is co-occurs in sympatry across much of the North American distribution of this species. And if we contrast that with nuclear genomic structure, again, you see northwest Mexico is kind of split out, but across this blue area, there's essentially no observable nuclear population structure, suggesting that these 2 mitochondrial lineages, which are about 2% divergent, are segregating on similar nuclear backgrounds. And so I'm curious about how has this apparent mitonuclear compatibility been maintained through the history of the species. And I've kind of broken it into 2 main hypotheses, which are probably not completely exclusive, but one is that mitochondrial DNA is just functionally neutral. These 2 haplotypes are not impacting fitness. The other is that you may have compensatory nuclear evolution. So these nuclear encoded proteins may be under selection to keep up with the divergence we see in the mitochondrial genes. And we might expect compensatory evolution to more likely occur if we have some segregation of these mitochondrial lineages across ecological gradients, or if there's evidence for relaxed or even positive selection on these mitochondrial DNA, I would expect nuclear compensatory evolution. If there's amino acid substitutions that are altering protein functionality, or if there's, finally, you would expect evidence for divergence in nuclear mitochondrial genes in association with the different haplotypes. So to test these predictions, I sequenced 235 individual savanna sparrows from across their distribution using an exome capture approach, and another 87 individuals I had whole genome sequencing for. And from all these individuals, I was able to extract 283 whole mitochondrial genomes using this method, novoplasty. And I'm not going to get into the nitty-gritty of the details, but you can ask me, or bioinformatic details, but you can ask me later. And with this data set, I essentially wanted to ask, how does mitochondrial haplotypes vary spatially? Are mitochondrial genes characterized by greater intensification or relaxation of purifying selection? Are mitochondrial substitutions likely to impact protein functioning among these different haplotypes? And is there evidence for divergence in nuclear mitochondrial genes in association with these different mitochondrial lineages? So to get this first question, I want to first show what the nuclear population structure looks like in this genomic data set. So if you look at this principal components analysis here, you can already start, and I'm mostly ignoring the birds in Mexico for this talk and focusing on the nominate clade, which is what's shown in this principal components analysis. And you already start to see a little more structure than we had with RADseq data. So you start to pull out some of these Alaskan birds and these Pacific Coast birds, and then everything else is kind of in the middle. And on the left, you can see an effective migration surface for this species. And it roughly shows similar patterns to the principal components analysis, where you get a lot more isolation on the Pacific Coast populations. But from Alaska, dark brown is more isolation, blue, more connectivity. And you get more of a blue shading across Alaska and Canada, suggesting there's widespread connectivity across that distribution. Looking at how mitochondrial structure looks across this distribution, here just using a neighbor joining tree to quickly cluster all these mitogenomes, you see the same clades, the blue and purple, the B and C clade from earlier. And then the A clade actually looks like we have a fourth subclade there, and then I'll get into that a little more. And then it just did time-calibrated phylogeny to try to understand when these lineages started diverging. So the B and C clade diverged from A starting about 600,000 years ago, and within B and C about 400,000 years ago. If we plot the frequency of these haplotypes across North America, we can start to see that there may be some latitudinal patterns in the frequency of these different haplotypes. So especially if we split A into two subclades, you see with the subclade A1 in pink and A2 in red, there is significant increase of A1 and this B haplotype as you move north, with the A1 haplotype pretty much only found in Alaska and other northern latitudes. So next I wanted to see if these mitochondrial genes within savanna sparrows are characterized by changes in the intensity of purifying selection. And to do this, I use this program, RELAX, which very briefly, you basically are comparing a set of reference branches on the phylogeny to a set of test branches and estimating this parameter K, where K greater than 1 is suggestive of intensified purifying selection, whereas K less than 0.5 is suggesting a relaxation of purifying selection. And so to run this, I also got a bunch of mitochondrial genomes from a bunch of other sparrow species to serve as my test branches, as my reference branches, and the savanna sparrow lineages to serve as test branches. And I did this for each of the 13 protein coding genes in the mitochondrial genome. And the results can be seen here, where along the x-axis you have each of our protein coding genes, and K is on the y-axis, and that dashed blue line is K equals to 1. And so you can see some genes have a parent intensification and some seem to be relaxed, but none of these results were statistically significant, suggesting savanna sparrows basically have the same level of purifying selection as most other sparrows. All right, so next I want to explore how the mitochondrial substitutions differing among these mitochondrial genome lineages may actually impact protein functioning. And so I looked at fixed amino acid substitutions among these different 3 mitochondrial lineages. Along the x-axis, again, you have the 13 protein-coding genes, and then the proportion of substitutions on the y-axis. And so the green triangles are comparisons between those 2 A subgroups. And you can see there's essentially no amino acid substitution differences between those 2 clades, with most of the amino acid substitutions occurring between B and either of those A. And so there's a total of 27 substitutions, and the next step I want to do is try to understand whether these would be predicted to alter protein functioning. So as a first pass, I used this program SIFT, and we see that only 3 of the 27 substitutions actually are predicted to alter protein functioning. So it's not necessarily completely neutral, and I'd like to follow up on some of these. And one way I'm going to do that to start is just look whether there's evidence for divergence in the nuclear interacting mitochondrial genes across these different lineages. And so just as first pass, I've looked at FST divergence between these different mitochondrial lineages. So this is between the B lineage and the A2 lineage, and this is classic sort of Manhattan plot where you have chromosomes on the x-axis, mean FST on the y-axis. And there are some peaks there, and some of these peaks are associated with known mitochondrial interacting genes encoded by nuclear mitochondrial interacting genes. None of these are associated with the most divergent peaks. And if we look to see if this is more than we expect by chance, it's not. So there are a couple intriguing ones I'd like to follow up on. But I also did the same thing comparing the B to the A1, where these 2 lineages segregate within Alaska. And same thing, you can pull out a couple genes, only one of these, which is overlapping with the previous Manhattan plot. And again, this is no more than you would expect by chance. So just to wrap up real quick, revisiting our predictions, well, we do start to see some associations between these mitochondrial lineages and latitude. The bulk of the evidence seems to point that we don't really have a significant change in purifying selection. In general, mutations are not altering protein functionality, and there's really not strong evidence for widespread divergence in nuclear mitochondrial interacting genes. And so this is really ongoing analyses, and I hope to look further into like tRNA divergence and rRNA divergence among these lineages across the mitochondrial genome. But I just want to conclude by kind of zooming out and trying to understand the evolutionary context of these savanna sparrows are diverging. So savanna sparrows and I think it makes sense why we might expect more functional neutrality among these lineages within the species. So savanna sparrows have an enormous range. I've shown previously that they have a pretty high effective population size relative to some other sparrows. 2% mitochondrial divergence isn't particularly large, I would argue. And birds tend to have lower mitochondrial substitution rates relative to mammals, and especially relative to some of these other lineages where you do see lots of mitonuclear coevolution, like the Selene plants, which have very high mitochondrial mutation rates. Or you have cases like this eastern yellow robin in Australia, where you have pretty deep divergence between mitochondrial glades across steep ecological gradients, or small populations where you have accumulations. All right, I'm going to end there and let the next person go.
Hi everyone, my name is Andy Tan, and I'm a second-year PhD student from Jing-Chun Lee's lab at the University of Colorado Boulder, at which our lab studies symbiosis in marine invertebrates and the underlying genomics. Today I'll bring you on a tour into the genomic world of sunbathing giants, and that will hopefully make sense in a moment. So there are more and more compelling studies that shows us the benefit of using long-read sequencing technologies such as PacBio and Nanopore, because they tell us more about not just how genes relate to one another in terms of their positions more completely in genomes, but also tells us a lot of hidden genomic structures and architectures that were previously not seen. And some of these technologies also challenges some of the more classical traditional view of how we perceive gene and genome, how they interact with one another. And more importantly for this talk, it calls us into reconsidering some of what we consider complete published mitogenomes online in GenBank, for example, whether or not we should reconsider how complete they are. So these are archetypal examples of what we consider as conventional mitochondrial genomes. These are examples from humans and sea snakes. Like in vertebrates, we often assume that they are highly conserved. Across metazoans, we see that the gene order is tightly ordered, deeply conserved, and in this case, you can see that they are almost entirely identical. And conventionally, we find the 13 mitogenome genes that are involved in respirations, and then we tend to make sure that we have all of them. We have all the tRNAs, rRNAs, and then we just tie the two ends together, and then you have your circular mitogenomes. But recently, there are studies that shows that maybe this method of just checking the 13 genes is not what is actually going on in the mitogenome. And so in this example, in this schematic, you see like when you have a long read, you get to see the details of like repeated sequences. We get to detect presence of repeated sequences. And without long reads, if you only use short reads, you fail to accurately assemble that region of highly repeated sequence. And in our case, because of our involvement with the Aquatic Symbiosis Genomics project in our lab, we managed to get our hands on long-read, high coverage genomes of our study organism. And at the same time, we ran a side-by-side Illumina sequencing de novo assembly with only short reads of like 150 base pairs Illumina sequencing, and we failed to recover some of the regions that turns out to be quite long. And this discrepancy in mapping and assembly has been reported in other studies too. So for this, as an example, the Vertebrate Genome Consortium, Vertebrate Genome Project Consortium in 2021, they assembled 100 mitogenomes of vertebrates using long reads that stretches beyond known genes, and they found out that they have widespread repeats and gene duplications in more than half of the vertebrate that were previously sequenced. But now when they redo the mitogenome, they find that there are new things to discover in their mitogenomes. And since mitochondria is involved in respiration, our lab thought that there is one particular group of marine mollusk that has a unique relationship with oxygen too, which is one group of marine bivalve from the family Cardiidae. There's about 270 species of them, and more vernacularly they are known as cockles. And in particular, our lab studies one group that are practically farmers of the marine realm in the molluscan world. They host these photosymbiotic dinoflagellates known as Symbiodiniaceae in their gills. These are the two groups. One group is a subfamily called Tridacninae, more commonly known as the giant clams, and then the other group is the hard cockles called Phraginae. So these are the organisms that I will be covering in this study today. And on the top, you will see Phraginae. It is, on this tree, everything in green are the photosymbiotic cockles or clams, and then the one in orange are the non-photosymbiotic species that we have. We have the complete mitogenome as well as the genome, but today I'm only covering the genome part of the study. And Phraginae is split into two clades nicely, which is the photosymbiotic clade and the non-photosymbiotic, but unfortunately today I don't have any examples of the non-photosymbiotic clade, but Tridacninae has two genera and all of them are photosymbiotic. I also have three other non-photosymbiotic cardiids as an example to compare their mitogenome to. And so we asked two questions of these clams. Do they have a non-coding region in their mitogenome? And if yes, what that could mean. And I'm going to briefly cover how we can easily detect whether they have repeat patterns in their mitogenome. And so this is a dot plot example. It's just a random example that I chose from a mollusk. And then when you map the mitogenome against itself on both axes, you get this clear diagonal line. This shows that it's an exact match. You don't have any other architectural like repeat patterns. It's just a clean one-to-one relationship. And when you have repeat patterns, you see this parallel lines along the main diagonal line. And this is often what you see as, when it's small, it's like a microsatellite or a macrosatellite pattern. And so now let's take a look at the non-photosymbiotic cardiates that we have in our analysis. You see these three very clean, strong diagonal lines and with small microsatellite-like pattern repeats. But the picture completely changes when you look at the photosymbiotic ones in our study. You see very complex, huge region of repeat patterns within your mitogenome, and that we haven't seen in any other groups that we study. And so the answer is a resounding yes. All of them have a very extensive non-coding region for photosymbiotic cardiates, and then all of the non-coding regions have extensive complex repeat patterns. And now let's take a look at the gene arrangements within the mitogenome. We managed to find all standard 13 metazoan mitochondrial genes, which are shown here all in green and blue. And then rRNAs are in brown, tRNA in yellow, non-coding regions are noted in red. As you can see here, there are high levels of gene rearrangements within the mitogenome of these cardiates, which has been reported in bivalve to be like there you see a higher level of gene order rearrangements, but it's still worth noting that it's quite high, even though they are sort of like constrained within genus. These are the positions of COX-2. Just some example, this is how ND4L is placed in like 5 different positions within this family. And you can see here, the non-coding regions are actually placed in at least 4 different positions within the entire family, even though they are restricted to like one genus having one position. So it's either that the non-coding regions arise independently or they have been moved quite rapidly throughout the entire family. And so what are the possible biological implications that we can tell from this group? Well, in some annotations in the non-coding regions, we don't find any known genes so far. But for example, in Phred here on the left, you see that we managed to annotate 11 copies of like extra tRNAs that were previously already in the coding region, but we managed to find them in the non-coding region too. And I'm going to show you how extensive that non-coding region is in comparison to the entire mitogenome. So here, highlighted in red, these are the portion of the non-coding region in relation to the entire mitochondrial genome. That these are for the photosymbiotic cockles, and when you compare it to the non-photosymbiotic cockle, it's much smaller. And here's some summary statistics. Even though I only have 10 samples, 10 species, you can see the stark difference between all the orange bars. You have non-photosymbiotic cockles compared to photosymbiotic cockles. In terms of mitochondria genome size, you see a range from 22,000 base pair all the way to a whooping 92.7 thousand base pairs, which is 5.8 times higher than like your typical metazoan 16,000 base pairs, which is much, much higher. And then the non-coding region also ranged from 6,000 all the way to 76,000 base pairs, which is very extensive. It accounts up to 80% of the entire mitogenome, which is a lot when you think about if 25 to 80% of your mitogenome is technically coding for nothing useful, then that is a lot of base pairs to be carrying around in your mitogenome. And I got curious, I also started looking at ORFs, which are basically sequences that could potentially code for a protein or RNA or any other elements that can be translated. We find that ORFs are present all throughout the coding and non-coding region, and here you can see shaded in green, those are the non-coding region, and you see basically an even like amount of ORF throughout. And I also got interested in looking at, I started masking for transposable elements and transposable element fragments within the mitogenome, and we also see the similar pattern. You have increased ORF as well as increased transposable elements that can be found within the mitogenome of both, especially for photosymbiotic ones, you have a much higher number. And here is, the followings will be like representations of like the gene annotations that I made in a linearized manner. And for all the samples you see from now on, it will be genes followed by detected tender repeat patterns, and then ORF that matches with like TE fragments or TE that I managed to mask. And here are all for non-photosymbiotic, and you see that there are, if you look at the blue boxes, there are not that many repeat patterns. But when we compare this to a photosymbiotic one, within the blue boxes, you see high levels of repeat patterns as well as high numbers of ORFs that matches TE fragments. And the same, it goes to Fragine. And I want to bring your attention to the last sample here, which is the one with the 93,000 base pairs. And you can see it's extensively covered in TEs, and up to 60% of it is covered in TE. And this bewildering animal grows up to an immense size of just 5 mm, which is that tiny dot up there, compared to like the other organism that we have in our study, which is the Tridacna gigas, the giant clam. It is the largest living shelled mollusk today, but that small thing actually is the smallest sample that we have, but it has the biggest mitogenome so far as we can tell. And so in a clamshell, there are long NCRs within photosymbiotic cockles with high numbers of repeats, high tRNA copies, high levels of ORFs with transposable elements that matches. We think that all these conditions could be associated with photosymbiosis, mainly because there could be sites for novel gene predictions, there could be oxidative stresses that leads to this relax in selection for the ability to regulate their mitogenome size. That could also be suppressed immunity in order to allow for symbionts to coexist in their environment. They have to suppress the T regulations. So yeah, so maybe what we consider complete might not be as complete, and thank you.
Can mitochondria drive nuclear gene expression? My name is Federico from the University of Bologna, Italy, and today I want to speak with you a way with which mitochondria may in some way drive nuclear gene expression. Generally speaking, there are many signals that are sent from mitochondria to the nucleus that may take several aspects like ions, catabolites, ROS, as was said before, small peptides, long RNAs. So there is a lot of stuff that mitochondria can use to communicate with the nucleus, including small RNAs. And actually, Andrea found some years ago in the Manila clam Ruditapes philippinarum such a small RNA in the mitochondrial genome of both male and female Ruditapes philippinarum. So they were found mostly in non-coding region, small non-coding RNAs that had nuclear targets. But why did we look in that species and not in another species? This is because Ruditapes philippinarum, the Manila clam, is a Dewey species. Dewey means that essentially both paternal and maternal mitochondrial DNAs are maintained and kept separate. So Dewey means doubly uniparental inheritance. Essentially what happens is that both sperm and eggs deliver the mitochondrial DNA to the zygote, and so the zygote is essentially heteroplasmic at the beginning, and then what happens depends upon the sex of the embryo. If the embryo is female, male mitochondria, paternal mitochondria are generally destroyed, and so the adult female will have only maternal mitochondria, both in the somatic line and in the germ line. But if the embryo is male, so paternal mitochondria are maintained and are kept only in the male germline. So the male adult will have paternal mitochondria in the germline, and so in the sperm, and should have only maternal mitochondrial DNA in the somatic line, but there can be different degrees of leakage. In this way, the original condition is restored, since sperm will only have paternal DNA, mitochondrial DNA, and eggs will only have maternal mitochondrial DNA. So the question was, is it possible that mitochondria, at least in this species, in some way drive sex determination? So we started looking from Manila clam exactly because we were looking for a way for mitochondria to regulate the nucleus, to tell to the nucleus, to the zygote, to the embryo, you must become a male, you must become a female. And so the idea was that, let us look for small non-coding RNAs. And as I said before, we actually found. And these were some of the targets that were using a bioinformatic pipeline predicted. And as you can see, many of these, I'm sorry that I was pointing on my right, but it's very difficult to, I have just one hand. Okay, here. And you can see that many targets are involved in sex determination, or at least are homologous to proteins that in humans are involved in sex determination, or are involved in cytoskeleton moving, so this may explain mitochondrial movement, or are even involved in methylation and acetylation, so it is possible that there is a regulation on the nuclear genome, a nuclear-wide regulation, and so on. But are they true? I mean, these were only in silico clues, but do these small RNAs really exist? We call them Smith RNAs, standing for small mitochondrial highly transcribed RNAs. And we have many clues of functionality. The first clues I can show you is conservation. Most Smith RNAs map on non-coding region. However, the region where they map are much more conserved than other non-coding region of the mitochondrial genome. So this is a strong clue for a selective constraint on that region. And this is true for many different Smith RNA that were investigated. Please note that we are still within the Manila clam model system. And then we carried out some in vivo experiments where we experimentally injected Smith RNAs into clams, and we had control clams, and we could demonstrate that the level of acetylation and methylation, for example, was significantly different in injected and in non-injected individuals. So this calls for a real effect of these molecules, again, in the model system of the Manila clam. And finally, we started looking outside the Manila clam, outside Ruditapes philippinarum. We looked in other metazoans, and it turned out that using the same bioinformatic approach, we always find candidate Smith RNAs. So, for example, I show you here my results on the zebrafish, on the fruit fly. We found different numbers of Smith RNAs in males, in females. Actually, currently we have a much more structured pipeline to do that, and we are applying that on many more species. Probably it's pointless now to list all the numbers that we found, but the point is that in every metazoan we search in, we find Smith RNAs. In some cases we find a few, a handful. In some other cases we find many, but we generally always find. So Smith RNAs are small regulatory RNAs that are produced by the mitochondrial genome that have nuclear targets that seem to be actually widespread, at least in the animal kingdom. So, if we want to explore the biogenesis and the, well, let us say, the working mechanism of these molecules, we have to try to understand how they become mature and how they can exit the mitochondrion. Exiting the mitochondrion is not a trivial task because mitochondria have two membranes that are very different and so on. Well, at first in Manila clam, we have many ways to do that. For example, it is known that at least in clams, mitochondria can open out and the matrix content is poured into the cytoplasm. So we can find extruded cristae in the cytoplasm. And many proteins that are normally involved in small RNA maturation, like argonaute, for example, in this case, can be found associated to mitochondria, for example, in humans. So there are clues of connectivity between the pattern of small RNA maturation, one side, and mitochondria, the other side. Generally speaking, we recently investigated a bit the many ways to trespass the double membrane system of the mitochondria for small RNA molecules, and we spotted out many different carrier systems that we consider as candidate for the carrier for these small molecules. PNPAs, the MPTP, the TIM-TOM complex, and even OXPHOS complexes that are known to be exapted to do other jobs than electron transport chain. All these complexes are involved in RNA translocation. Normally, they are well known to be involved in RNA translocation from outside to the matrix, from the intermembrane space to the matrix, from outside to inside. But there are clues, or at least it is conceivable, that they can work the other way around. So this is still to be explored, but there are many possibilities, I mean, for these molecules to escape the mitochondria. What is particularly interesting in our opinion about these small molecules is their, I mean, great evolutionary potential. If it is true, and we prove that at least in the Manila clam it is true, a small molecule, a small RNA which is produced by the mitochondrial genome can affect the nuclear genome acetylation and methylation. This means that by a very simple molecule, the mitochondrion has the potential to, at least in some condition, at least in some species, turn on and off large section of the nuclear genome. Which is the way, for example, it could influence sex determination in the Manila clam, where we started from. And it is important to remember that these molecules are very, very, very small. I mean, the mature molecule can be 22, we normally approximate them. We are trying to understand the current, the actual biogenesis, but we approximate them with the length of microRNAs, so 20, 22 base pair. So it's not a matter to be humble and small things can do great and pride and so on. The point is that it is very easy for them to evolve de novo. It is very easy to conceive that they can emerge de novo. And actually, there are many examples, for example, in Drosophila, of de novo emerging microRNAs. And we run some simulations, and we show that the probability of finding, completely by chance, a new target in the transcriptome is quite high and is maintained in different species. So it is very probable that if a new molecule like that emerges in the mitochondrial, by chance it will find a target in the nuclear transcriptome, and then natural selection will decide. If there is an adaptive issue, it will start to work on this new molecule and so on. Otherwise, it will be probably destroyed. And possibly, this is particularly true when we have a mitochondrial genome with a particularly prone to evolve new function. I mean, the human mitochondrial genome is typically very boring, very close, not so many functions and so on. But we know that bivalve are plenty of non-coding region, huge genomes, bewildering rearrangements, and a lot of many different secondary structure. So probably it is very easy to for a mitochondrial genome like that of bivalves, to evolve genes like that completely by chance. And this was exactly the end of the presentation. I have to thank all my collaborators. Thank you very much for the attention.